Hi everyone, thank you for the nice introduction and for the opportunity to present my work. So you probably already know that the human bronchial epithelium is a well-differentiated pseudostratified epithelium composed of basal cell, secretory cell, and also ciliated cell, and it's resting on a connective tissue with interstitial fibroblast. And here in this conference, we are interested in drug delivery, right? And we want to understand how the drug can permeate to the mucosa. And to develop a 3D model that better reproduces cell interaction with the matrix would be a really useful tool to study this drug inhalation and also to study the lung biology. So the first 3D model used are monoculture of human bronchial epithelial cell grown at early in phase. They support epithelial differentiation, mucus production. They are actually really good model if you are interested in the epithelial part of the mucosa. It's a peak thickness model. But what about the connective tissue? Because we know that in many pathologies, the interaction between the fibroblast matrix and the epithelial cell is really important. So having a 3D model that properly reproduces the full thickness with bronchial epithelium and fibroblast would be really pertinent. And usually to make this kind of model, they used hydrogel or porous mucoscaffold. They are really useful, but they had some limitations, such as the contractility. So in this study, I tried to do a scaffold-free tissue engineering method using the self-assembly method. So meaning we use the cell ability to produce and organize their own extracellular matrix. And this method has been successfully used in many organs already, like skin, but so far have not been done in bronchial model. So these are the two models I'm trying to develop in this study. Let's start with the split thickness model. So we use the protocol that is already established. We use the BCI NS.1 normal bronchial cell line. We seed them on a porous membrane, and then we wait until they are fully confluent. Then we do what we call the air liquid interface. Then we allow them to differentiate for a few weeks until they form a pluristratified epithelium. The protocol provided by Ronald Crystal Lab actually recommend 28 days of culture. But since I knew that later on I was going to do a model that took even longer to grow, I tried to see if I could do a shorter time of culture, 21 days. Here what I got. So at 21 days, we can clearly see on the HT staining, we have a pseudostratified epithelium. With the past staining, we can see we have acidic mucin, with the Alcian blue, neutral mucin, and overall it really looks like a human oral mucosa, bronchial mucosa. And at 28 days, you can see it's thicker, less organized, with visibly more mucus. So I also measured the TER, and it was quite similar between 21 days and 21 days, so around 750, which display good barrier function. Then I tried to really assess the differentiation of that model. So I would do immunostaining, targeting keratin 14 basal cell marker, mucin 5AC mucus protein for the secretory cell, and FOXG1 nuclear marker for the ciliated cell. And when you do quantification, at 21 days, you have roughly 30% of each cell type. But at 28 days, you have 50% of secretory cell and only 15 of ciliated. So there is a shift between ciliated and secretory. And this was also confirmed by some image where you can see at 21 days, you can clearly see more ciliated cell and 28 more secretory cell. And also in fluorescence microscopy, when you look more into the mucin 5AC and 5B, you can notice at 28 days, the mucin 5A really start to appear compared to 21 days. And we can also distinguish that when you do the Z-stack, 28-day mucus 5A is much more present. So with those experiments, we decided for the next in vitro model to keep the 21 days time of culture because we had a better repartition with secretory and ciliated cell. So second model, the full thickness model. Basically, apart from the cell, you only need a paper ring made out of filter paper. Bottom of your well, you put your fibroblasts, we use fetal lung fibroblasts, then you cultivate them with vitamin C, ascorbic acid. And it will promote the production and the organization over the extracellular matrix. And then after 3 weeks, the fibroblasts will preferably attach to the ring paper instead of the bottom of your well. That's when you know you can seed your epithelial cell on top. And when you have full confluency, you do the air liquid interface. So you use a 3D printed Ali support, and then we use 2 paper ring with only fibroblast and matrix, and then one with the fibroblast and the epithelial cell. We stack them, we wait 21 days, and we see how they differentiate. And here you can see, so on the picture, you can really see the fibroblasts embedded in their own matrix are really tied to the paper ring. So you can manipulate them freely. And then you look at the HT staining. So we have the epithelial part, and then here you have the fibroblasts embedded in their own extracellular matrix. And I want to remind you that all the matrices you see here have been done by the fibroblasts. Nothing has been added, no collagen. And then you look at the pus, you have like acidic mucin. With the Alchem blue, you also have neutral mucin. And when you try to also assess the differentiation, again with the same marker, basal cell, secretory, ciliated, you have around 30% of basal cell, 15% of secretory, and 36% of ciliated, much more than in the split thickness. Also with the same, we can really see number one here, your secretory cell, and then in 2 and 3, the cilia and the mucus film. So those are my two models, but then I want to use them for drug delivery, so I need to be able to do permeation study, right? So the first one we did was liquid-liquid permeation using salbutamol, and also 2 integrity markers, FD20 and Lucifer yellow. And when we look at the TR after the liquid permeation, after 6 hours, it doubles compared to before. Almost as if having liquid on top of the sample modified the resistance, or maybe the liquid and the salbutamol. And then we try to see the permeation of salbutamol, and we can see there is a slow increase until 6 hours. So that means that the BCI are partially permeable to salbutamol. But if you look at the integrity marker, whether it's FD20 or Lucifer yellow, the amount permeated is less than 2%. So that means that you still have a good barrier function. So then we try to do air-liquid interface permeation to see if you could have some effect on the transepithelial resistance. So the air-liquid permeation, we use a device that was already published. So it's simply 1 ml syringe with 0.5 ml of air volume, then a valve that we close so we can increase the pressure. You open the valve, or here we go into a tip in which you have the loading powder, and will be dispersed into a cone made of Eppendorf tubes directly on your sample. And we did it both split thickness and full thickness. For the TR of the split thickness, we could see that after the permeation, we had roughly the same value, meaning the early permeation did not affect the resistance. But for so these are still preliminary data, but for the permutation of salbutamol, we had the same from split thickness to full thickness. But it was 5 times higher than on liquid permeation. There is maybe two explanations to that. First one, it had been shown that at early permeation is higher than liquid permeation. Second one is because of our protocol. Here we have 0.5 milligram of powder, and we know we have approximately 50% of that powder that actually end up on the sample. And it's much higher than in the liquid permeation where we had like 120 microgram of salbutamol. So in conclusion of this study, we have a first split thickness model that is capable of differentiating in a pseudo-stratified epithelium with ciliated, secretory, and intracellular mucin. And we know that the salbutamol partially permeates through the epithelium, but the TR value are still correct. And we have an integrity marker below 2%, which provide good barrier function. Then we have the full thickness model, a scaffold-free model that prevents the contraction of the fibroblast. And the good thing is that the lung fibroblasts were able to respond to the vitamin C stimulation and produce their own extracellular matrix and organize it. And also our epithelial cell were able, on top of that matrix, to differentiate. So it's a really good in vitro model with really low material and logistical cost and high reproducibility. And for the air liquid permeation, we use a really simple, effective device for the powder dispersion. And the salbutamol micronized dry powder permeation was similar, both with split and full thickness model. So in this presentation, I talked a lot about the epithelial part and not many about what's happening in connective tissue. So that's what's coming after. We want to try to see if there is any crosstalk between the epithelial cell and the fibroblast. And also, this is a Hershey model. So on the longer term, we want to try to induce the pathology. Or even use cell from patient. Thank you for your attention, and we're happy to reply any question.
Thank you very much for Aurelie for this very nice presentation. Are there questions? Please come up to the microphones, or the microphone will come to you. Just wave, and we try to see you. It's always a bit difficult, but there is a first question. Thank you.
Coming half the way to the microphone. Sorry for my voice. Carsten Air, Trinity College Dublin. Having a barrier is nice and fine. What about active transport or generally membrane transporters in the model compared to native epithelium?
Sorry, can you repeat, please?
Membrane transporters. Is the expression similar, or have you looked at the expression?
I don't know. That's something we should check if it's like, for example, the same as the Calitrice model that everybody uses.
No, it should definitely not be the same as.
Yeah, it won't be the same because the permeation is higher.
Yeah,
but yeah, that's something we should check.
Okay,
yeah.
Thank you.
So maybe while you think about further questions, I have a question for you. And this basically lines up with the previous one. Did you do already any comparison between your model and other established models to see differences or maybe advantages of your model?
Yeah, so we did comparison with Calitrice model because we have it already in the labs. So on morphological way, the difference is that Calitrice is monolayer. They have a lot of ciliated cell, a bit of mucus, but it's a monolayer. It's not a pseudo-stratified epithelium. On a permeation side, Calitrice are much more tight. And for example, salbutamol barely penetrate, but we have much more. We have more penetration. So that's something, yeah. Also on the TR, for example, Calitrice, when you do permeation over like a certain amount of time in liquid permeation, the TR does not change before and after the permeation. While our Calitrice, we don't know, the BCR, we don't know exactly why TR increased. That's also one of the change, yeah.
There's a great talk, thank you. I was just going to ask, have you tried the 28-day ALI culture on top of the fibroblasts, see if there is a difference in that organization that you were seeing?
Sorry, can you repeat, please? We did not understand.
I was asking, have you tried the 28-day ALI culture on top of the fibroblasts to see if there's a change in that organization that you were seeing?
Yeah, no, I haven't tried. That's a good question. It already take 43 days with 21 days, which is long. So yeah, I think I will try to do it. But what I'm a bit afraid is that my epithelium is quite thick already. And I think it's because of fibroblasts, because it's thicker at 21 days in full thickness and in split thickness, even if we have the same concentration of cell. So I think I will try, but I'm a bit afraid I will have something like in the split, like something really thick, really disorganized, and with a lot of mucus. So yeah, that's something I'm planning on to do, but I haven't done it yet. Yeah.
All right. I screen through the auditorium again. Any other question? There is one. Microphone is coming. Okay, wasn't the question gone already? So I would like to thank you again for this very clear presentation, and we move on to the next speaker. Thank you again.
Thank you very much.